Hello, everybody, and welcome back. And today, now we will be talking about the proteostasis crisis. Okay, this is a very interesting module. It's a little bit complex, but please bear with me, okay? So, the proteostasis crisis, as the definition says, the term refers to the disruption or imbalance in proteostasis, which is the maintenance of protein homeostasis within cells. Now, proteostasis involves the proper folding, assembly, trafficking, and degradation of proteins to ensure their functionality and prevent the accumulation of misfolded or damaged proteins. Now, we wouldn't want to be having, too many proteins that are damaged or misfolded building up in our cells, because this would lead to pathology. This would lead to disease, and I'll show you an example in just a little bit. So when proteostasis is dysregulated or it's impaired, it can lead to the accumulation of misfolded proteins and protein aggregation, which ends up causing cellular dysfunction. Okay? This is a hallmark feature of various neurodegenerative diseases, okay, as Alzheimer's disease, Parkinson's disease, Huntington's disease, amyotrophic lateral sclerosis, and we'll talk in particular as a model example with Parkinson's disease. A proteostasis crisis may occur due to various factors, including genetic mutations, such as in Huntington's disease, environmental stressors, okay, aging, and metabolic disturbances. And when I refer to metabolic disturbances, usually I'm referring to disturbances that affect our microbiome and microbiota, which can actually affect all of these, as a matter of fact, okay? So let's go ahead and deepen down a little bit more, okay? So here we have our cell. Here's our nucleus, right? Here we have our endoplasmic reticulum, and it has its ribosome. So this is going to be the rough endoplasmic reticulum, right? Here we have our Golgi apparatus. Here we have our mitochondria, okay? And the rough endoplasmic reticulum, which has the ribosomes, that's the reason why it's called rough endoplasmic reticulum. It's always usually found near the nucleus, okay? As you can see right there. This is the nucleus, it's right beside it. Now, the main function of the rough endoplasmic reticulum is to be able to make proteins. So it participates in protein synthesis. And then once these proteins are made, it gets sent out and transported to the Golgi apparatus, okay? When here, the Golgi apparatus is going to be activating or eliminating specific proteins, all right? And if it's going to be activated, it's going to be later transporting these into vesicles, which eventually are going to be taken to the cell wall and probably eliminated through a process known as exocytosis. Now, this process can also be done through endocytosis, where the cell takes in certain proteins and it puts it back into the machinery, right? Or if in the case the Golgi apparatus is going to be eliminating some of these proteins, it will be doing it through a organelle known as the endosomes, okay? All right. Now, the smooth endoplasmic reticulum, which does not have ribosomes, usually this is found near the membranes, and its main component here is participating in the production of lipids, so it participates in lipid synthesis. But we're going to be focusing mainly on this portion right over here, which is the rough endoplasmic reticulum, okay? So once again, it's very important to remember that proteostasis involves folding, assembly, trafficking, and degradation of proteins, okay? These are four important things that must take place, all right, within the production of proteins, okay? Now, when this rough endoplasmic reticulum is not working correctly, we start having issues with the folding, the assembly, the trafficking, and degradation of proteins, and that is going to cause a series of problems. And as we can see right over here, all right, this is a post-mortem image we can see right over here, where we can find the medulla oblongata, where we have here the substantia nigra, okay, where dopamine is made in a healthy patient. This dark region right over here is full of specialized cells that are responsible in the production of dopamine, and this is known as the substantia nigra. And this would be a histologic image of a healthy person, for instance, right? But in the case of Parkinson's disease, and we know that Parkinson's disease is actually an autoimmune condition, we start seeing that they start developing what they call Lewy bodies, okay? And this is actually something that is very, very it's pathognomonic of patients who have Parkinson's disease. And as you can see, with time, we start losing this substantia nigra, and we start having difficulty being able to produce dopamine. All right? So that's what makes the disease very characteristic, is because the substantia nigra is now compromised, because we start developing what we know as Lewy bodies. Here we have a Lewy body, which are abnormal aggregates of proteins that develop inside nerve cells, and primarily in the brain. They are primarily composed of protein called alpha-synuclein, which forms clumps of aggregates within the neurons. Okay, so basically, this protein synthesis pathway has been compromised, and it starts building up this form of protein in the form of aggregates. Okay. So just to make things clear about alpha-synuclein, this is a protein that is very abundant within the human brain, and especially within neurons. It is primarily found in the presynaptic nerve, right, which is the communication points within another neuron, right, where the neurotransmitters are released, okay? And the normal function of alpha-synuclein is believed to play a role in the regulation of synaptic function and neurotransmitter release. It may also be involved in the maintenance of neuronal integrity and plasticity. So it's very important that these proteins, known as alpha-synuclein, be in its natural state. It doesn't suffer aggregation, okay? Okay, so in its normal state, alpha-synuclein adopts a mostly unfolded, right here you can see it, unfolded or disordered structure. And the more they start to aggregate, okay, then you start developing what we know as Lewy bodies, okay? So this starts leading to the formation of protein clumps known as Lewy bodies, as we saw in the last slide, all right? Now, post-mortem analysis of Parkinson's disease neuropathology have revealed that alpha-synuclein depositions, not only in the brain, but also in the spinal cord, the autonomous nerves, the peripheral plexus, the enteric nervous system, and in the nerves of the skin, submandibular glands, and myocardial tissue can also start to accumulate. So this is the reason why we start seeing issues not only with movement, but also we can start seeing issues with the enteric nervous system. So lots of these patients, for instance, who have Parkinson's disease have issues going to the bathroom, such as constipation. And the reason why is because they start building up these Lewy bodies. They also have issues with heart conduction, so they might actually have a arrhythmia. They might have lack of saliva because they start building up these Lewy bodies in their submandibular glands. And they may have issues with the skin, such as eczema, atopic dermatitis, or issues like, seborrheic dermatitis. They have a whole bunch of different manifestations that are actually being caused by the accumulation of these misfolded proteins known as alpha-synuclein aggregates, okay? Now, one thing that has been noticed through microbiome testing is that patients who have Parkinson's disease have an overgrowth of this bacteria known as Desulfovibrio bacteria, okay? And Desulfovibrio bacteria ends up producing what we know as hydrogen sulfide, which can also cause constipation. And also, obviously, they have lipopolysaccharides, which are endotoxins that can cross the gastrointestinal tract, causing leaky gut syndrome and affecting your immune system, but also can cross the blood-brain barrier. They also make small-chain fatty acids, obviously, but they also make indole compounds, and they make what we know as trimethylene N oxide. Okay? Now, trimethylene N oxide is produced in the gut by gut bacteria by dietary sources such as choline and carnitine and some other nutrients, for instance, but this mainly derives people who like to eat a lot of meat, okay? So when we have elevated levels of trimethylene N oxide, all right, they have been associated with an increased risk of neurodegenerative diseases such as Parkinson's. Now, there are a series of mechanisms by which we have to try to keep things in check just to make sure that, these misfolded proteins don't continue to cause all this, cellular dysfunction. So when you accumulate hydrogen sulfide, lipopolysaccharides, indole compounds, trimethylene N oxide, you start affecting the rough endoplasmic reticulum, which affects the Golgi apparatus. And guess what happens? We start having issues with folding, with assembly, trafficking, and degradation of proteins, okay, within the cell. And this eventually, in the case of Parkinson's disease, will cause the aggregation of alpha-synuclein, as we just mentioned. All right. Now, as I also mentioned, we have different systems in check that are always trying to make sure that this folding, assembly, trafficking, and degradation does not occur, occurs in the way it's supposed to be occurring normally, right? So, for instance, chaperone proteins are also known as molecular chaperones because they assist in protein folding by stabilizing unfolded or partially folded proteins and preventing their aggregation. All right. So, for instance, chaperones such as heat shock proteins help newly synthesized proteins achieve their correct three-dimensional structure. So, in other words, chaperone proteins are very important in order to allow proteins to have their correct dimensional structure and allow for these to not go through a misfolding, right? Then we have another process that keeps proteins in check, which is known as the ubiquitin proteasome system. So this is a system, basically, that's responsible for the degradation of misfolded or damaged proteins, okay, within the cytoplasm and also in the nucleus. And the protein that's responsible for doing this is known as ubiquitin because it attaches to target proteins and are marked for degradation, making them easy to be able to recognize and taken out for degradation through the proteasome, okay, which is a protease complex. Now, we have another system as well known as the autophagy lysosome pathway, which is responsible for, the sequestration, the degradation of cytoplasmic components that are old and, they need to be taken out. Or also, they're responsible for taking out damaged organelles or the presence of aggregated proteins, okay? And how do they do this is through the formation of autophagosomes, which fuse with lysosomes, right? And where these can be degraded through lysosomal enzymes. Then we have another system known as the endoplasmic reticulum quality control system. I know, it's a big name, ERQC. Okay, so this system is basically responsible for monitoring protein folding and assembly, okay? And it makes sure that these proteins are transported to their destination correctly, and if there are the presence of misfolded proteins, these will be targeted and taken down for degradation, okay? And participates in conjunction with the chaperone proteins, okay? And some specific enzymes. Now, in the case of the Golgi apparatus, as we just mentioned previously, the Golgi apparatus is very important because it has what we know as the vesicular trafficking. So it's responsible for this part over here. See, we were talking about the folding, the assembly, the trafficking right now. So the Golgi apparatus is responsible for the trafficking, okay? And proper vesicular trafficking between the endoplasmic reticulum, the Golgi apparatus, and other cellular compartments. So, in other words, it's responsible for the correct transport of this protein into where it's supposed to be going to its final destination. Now, we also have what we call the molecular degradation pathway, which is, in addition to the ubiquitin proteasome system and the autophagy lysosome pathway, we have this pathway known as the endosomal sorting complex required for transport. It's just basically another pathway just to make sure that there is adequate degradation of certain misfolded proteins, okay? And finally, we have the protein quality control machinery, which are where we see the cells have specific proteins for quality control, and where it is involved in monitoring protein folding, assembly, and degradation, and also it's responsible for being able to recognize, repair, and remove misfolded or damaged proteins, okay, via regulatory mechanisms that coordinate with cellular responses to proteotoxic stress, okay? So the question is then, as you can see, there are many different mechanisms by which proteostasis is regulated by, okay? It's very complex, as I just mentioned earlier. But if all of a sudden you do a microbiome test and you see that there's an overgrowth of desulfo vibrio bacteria, and this patient is making hydrogen sulfide, lipopolysaccharides, indole compounds, trimethylene N oxide, and it starts affecting the folding or the assembly or the trafficking or the degradation, of the proteins in this case, we're talking about alpha-synuclein, right? How can we treat this? How can we actually fix this? Well, the first thing we need to do is that we know that the diet is the main way it's the main way how we can actually regulate how the microbiome is going to be modulated. So we put this patient on an anti-inflammatory diet, and we give them vitamin D to be able to reduce the intestinal hyperpermeability alongside with its cofactors. And in the case if there's just too much of an overgrowth, we might actually even introduce some form of antibiotic, herbal antimicrobials, such as oregano oil, berberine, allicin, you name it. And by doing that, we start reducing the population of this opportunistic bacteria, reducing all of these inflammatory substances, which will now preserve and allow the cell to be able to have adequate folding, adequate assembly, adequate trafficking, and adequate degradation, which leads to normal proteostasis and eventually restores normal cell function. Okay? Well, I hope you guys enjoyed this video, and I'll see you next time.